Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Matt Kuo from Wacker.
Hello, everyone. Good morning from San Diego. It is a pleasure for me to present here today, but before I do that, I would like to first introduce you to our company, Wacker Chemie AG. So Wacker is headquartered in Munich, Germany, and was founded in 1914 by Dr. Alexander Wacker. About 100 years later, there are now 26 production sites and 23 technical centers around the world. In 2020, Wacker reported sales of about 4.7 billion euro with profitability of nearly 700 million euro. You can also see the other associated investments listed here. Globally, there are about 14,000 employees, with around 11,000 of them in Europe, 1,600 in the Americas, and 1,900 in Asia. As you can see from the slide, Wacker has a long history with high financial stability. So Wacker has 4 corporate departments. They are the silicones, polymers, biosolutions, and polysilicon. Today, we'll be discussing the Wacker Biosolutions division, which is highlighted here in blue, and more specifically, the biopharma groups under Wacker Biosolutions. Under the umbrella of biopharma are 2 sites located in Germany, 1 site located in the Netherlands, and Wacker Biotech US here in San Diego, which is the subject of today's talk. Wacker is well known in Europe as the microbial CDMO with a solid track record and strong customer focus. That means we're able to develop processes for therapeutic candidates from scratch to market, and we have strong expertise in the production of proteins, live microbial products, vaccines, and now plasmid DNA. So briefly, I want to give an overview of Wacker's cutting-edge technologies. Our proprietary microbial technologies include several brands. Starting from the left is our Isotech system, which is suitable for secretory production of properly folded target proteins. There's also a second generation Isotech, which is just to the right of that. And then the next brand is Foltech, also an E. coli-based system. It is well suited for production of target proteins in inclusion bodies, which is subsequently followed by an in vitro refolding step, which results in correctly folded target proteins. The next one is Libotech, which offers a platform for GMP production of live microbials. And as already mentioned, we offer GMP manufacturing of vaccines. And finally, on the far right is plasmid DNA manufacturing, and that represents the expertise present here at the San Diego site. And it is the newest addition to the Wacker Biopharma portfolio. So Wacker Biotech is distributed globally across 4 GMP facilities spread across Europe and the United States. As you can see on the right-hand side of the slide, the sites in Halle in Germany and Jena, both located in Germany, are mainly focused on the production of recombinant proteins. And this also includes feasibility studies, process development, process characterization, cell banking, and GMP production. In the top left corner, the site in Amsterdam has a strong expertise in the fields of live bacteria, vaccines, fill and finish, and GMP production of proteins. And finally, on the bottom left, the San Diego site has a strong background on the production of plasmid DNA. This site, it's been in existence since 2003, and Wacker acquired this site earlier this year, around February. The GMP scales will range from a 6-liter to 30-liter variable scale single-use line and the existing 500-liter stainless steel line. So As mentioned before, manufacturing of plasmid DNA has been performed in this very facility for nearly 20 years. So not only did the acquisition of this site add plasmid DNA manufacturing expertise to Wagner's portfolio, it also established a U.S. presence for CDMO activities, as all the other biotech groups are located in Europe. Much of the senior staff here have been here since the inception, since 2003, and have been responsible for the release of over 100 GMP pDNA batches for both commercial and governmental entities. These activities, they encompass the entire process chain, from the initial cell banking all the way to fill and finish of bulk drug substance and QC release. These products have ultimately been used either for DNA vaccine production directly or as starting material for various client applications such as viral vectors, CAR T-cell therapy, gene editing, or mRNA production. So on this slide, you can see a summary of our collective experience here in San Diego. As mentioned before, this site has released over 100 GMP batches encompassing greater than 10 different pDNA products in a variety of ranges, size ranges, and with a variety of E. coli hosts. And in the middle panel, you can see our process development capabilities. The plasmid manufacturing process is animal product free and RNase free. And we have cloning, transformation, process development, and optimization capability within our PD group. There are also a number of bioreactors, lysis skids, TFF skids, and chromatography systems available for the production of plasmid. And then in the right panel, you'll see our GMP manufacturing capabilities. 2 GMP lines will be available in early 2022, which we'll talk about later. And both lines have their own dedicated associated downstream equipment as well. So as mentioned before, we have a dedicated process development lab to support feasibility, optimization, technology transfer, and comparability with staff that have been working with plasmid DNA for close to 15 years. The lab is currently about 800 square feet, but we are expanding, and soon it will be around 4,000 square feet, along with the appropriate staffing. So Wagner Biotech is focused on the development objectives of the client through the various phases of the clinical drug development cycle, and we have experienced staff to guide the way through the process. So Wagner aims to be a one-stop shop for the development process of pDNA, but recognizes that a one-stop shop does not mean one-size-fits-all solutions. Our business development teams will work with the customer to generate customized work packages tailored to their unique needs. From GMP cell banking all the way through the final pDNA product release, there is a continuous quality assurance and analytical support throughout. As far as capacity, there are also dedicated fermentation, recovery, purification, and bulking suites with the within the GMP manufacturing areas, and there's a lot of room here for expansion. So just on this slide here, you can see our menu of QC release tests that can be selected according to your application. There are a number of assays available in-house and some via multiple methods. For example, assessment of purity can be done via HPLC, capillary electrophoresis, or agarose gel electrophoresis. The lab is directed by someone who has been testing pDNA for about 22 years. And there's also the capability of tech transfer and validation of methods, as well as stability studies. Meanwhile, we've got an on-site QA team that works closely with the manufacturing and QC groups to expedite product release. So over the years at this facility, we've developed a platform pDNA manufacturing process that is highly scalable. All scales, they use the same buffers, resins, and solutions as the full 500-liter scale. The scales, the yields and quality at the 5 mil scale are predictive of the yields at the 5 liter and even the 500 liter scale. And over the past year, the facility has been renovated to upgrade the 500 liter line to increase functionality and automation. Soon, in early 2022, a single-use 30-liter GMP line will come online, and we already have another modular clean room suite constructed for future GMP expansion of another single-use line. It just depends on what the customer demand is. So briefly, I want to show this slide. We offer a number of manufacturing options to suit customer needs. On the left-hand side in gray are our non-GMP offerings. Where it makes sense, single-use materials are used, but most of the offerings are multi-use in this category. On the right side in purple are our GMP offerings. So in early 2022, we will offer a 6-liter to 30-liter variable scale line that is fully single-use, and also a 500-liter stainless steel line. At the bottom, you can see the expected pDNA yield, but that, of course, is highly dependent on the characteristics of the plasmid. So whether it's milligram amounts of material for preclinical or early phase clinical, or gram quantities for phase 3 and beyond, we can provide work packages to fit your requirements. Non-GMP or GMP or single-use versus multi-use at various quantities, they're all possible within this facility. So briefly, just to demonstrate the scalability of the process, I'd like to show the results of a case study that we recently did. So here on this slide, you can see the results of a single plasmid manufactured across 3 different scales: 500 liter, 5 liter, and 5 mil. The bar on the top left shows the volumetric yield, which is milligrams of pDNA per liter of fermentation broth. And the bar graph on the top right shows the productivity yield, which is milligrams of pDNA per gram of wet cell paste processed. So both of these graphs show that for the 500 liter scale, the 5 liter scale, and the 5 mil scale, the plasmid yields are comparable across the board, accounting for scale. And the chart on the bottom of this slide lists a selection of quality control assays. As you can see, after confirming the identity of the plasmid at all 3 scales, the QC results are also agreed quite nicely. The A260, A280 ratios are identical. The purity is over 95% in all cases. The endotoxin levels are below 2 endotoxin units per milligram in all cases. And the residual RNA host contamination is under 0.1% in all cases. So as you can see, this case study demonstrates that the yield and quality data are very comparable, and that the process is scalable over a range of roughly 100,000-fold. So now I'd like to highlight one of our most recent customers, Helix Smith. Helix Smith, they've invented a DNA plasmid that encodes for a novel human hepatocyte growth factor named Ingensis. It is used as an active pharmaceutical ingredient to treat diabetic peripheral neuropathy, and it's currently in phase 3 trials. And in 2018, the FDA granted RMAT designation to Ingensis. And here in San Diego, we've been manufacturing it under full GMP compliant conditions, under campaign mode and multiple lots. So we have extensive experience with manufacturing and releasing Ingensis. So just to briefly summarize, we've got a highly scalable platform plasmid DNA manufacturing process. We also have in-house quality assurance and quality control to expedite product release, and we have capacity for GMP manufacturing available in the 500-liter stainless steel bioreactor, and then in early 2022, we'll have additional capacity for single-use GMP manufacturing between 6 liters and 30-liter scale. We've got several yield, quality, and regulatory options available, and we offer comprehensive services for plasmid manufacturing, QC testing, and QA release. And we're also expanding our process development, quality control, and GMP areas. So with this last slide, I'd like to finish my presentation and just show some of the notable customers from our portfolio that Wacker has supported in various phases, from feasibility through process development and optimization, up to GMP manufacturing, and also process characterization across the various sites around the world. And okay, lastly, yeah, thanks for your attention. You can contact us at info.biologics@wacker.com if you have any further interest, and we can open up the discussion now and answer any questions that you might have.
Okay, the first question is, what is your typical pDNA productivity in milligrams of pDNA per liter of bioreactor culture?
So roughly, well, it will of course depend on the characteristics of the plasmid, but roughly 100 to 200 milligrams per liter is what we can expect under our current GMP production system. Now, over the past year, we've had a lot of upgrades to the facilities, to the equipment, and to automation. So those we expect will increase the yields. And the other part is now that we're part of Wacker, there are a lot of microbial engineering, strain and vector experts that we can now have that we now have access to. So yeah, what I foresee is a continuous improvement of the pDNA manufacturing process here, and yeah, it should be a very powerful combination going forward. So right now, our process it prioritizes turnaround time. So our fermentation is very short. But now with Wacker purchasing us, they have a lot of experience with high density fermentation. Of course, those will increase the time in plant, but it's all up to what the customer wants, what the demand is.
And what does your capacity look like for the near future?
So for 2021, our GMP production of 500 liter is all spoken for right now, but it should open up in 2022. For the 500 liter stainless steel line. As far as the 30 liter line, we're expecting that in early 2022, but we don't know exactly when. We're going through commissioning stage right now. So the best thing to do would just be to check with our business development team or through info.biologics@wacker.com.
And is there potential for going beyond the 500 liter scale?
We've talked about it, but right now it doesn't look like the customer demand is there. But this is something like if there is interest, please contact us and let us know. We're still gauging customer demand for various scales. Right now, it didn't look like there was as much interest in that as there was for smaller scales. 30 liter, 300 liter looked more in demand.
And what is the timeline for GMP plasmid production at the 30 liter scale in the current scenario?
So that I don't want to comment on because I'm not in the manufacturing operations area, but please contact us and we can give you an appropriate work package. This, of course, it's going to depend on the starting point, whether it starts from an insert, a plasmid, a cell strain, MCB. So all those things will contribute to the duration of that timeline.
Okay, well, it looks like we're running out of time, but we have time for just one more question. If we didn't get to your question, we'll pass those on to Dr. Kuo, and he can follow up with you directly. So continue to type your questions in. But the last question for the webcast is, what is the difference between HQ and GMP pDNA?
So GMP pDNA will have a lot more QA oversight and documentation. It's going to be more expensive too. Those are the big keys. Usually, well, it'll depend on the customer, like the types of QC assays that are run as well. It may be a reduced subset for HQ. So this term HQ has been, it's been out there quite a bit. I think it's not super well defined right now. So just keep that in mind.
Okay, thank you, Dr. Kuo.
Thank you. Thank you, everyone.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.